Yes. Welcome, everybody. Welcome. If everybody could take their seats, and don't be shy, there's room at the front. So come and some of the people from the back row can come and sit towards the front and leave those chairs for when people come in a bit late. So yes, that's the back row. Please come and move up to the front a little bit. Okay, thank you. And I'm so pleased to see everybody here tonight. So welcome. And most importantly, I would like to recognise and pay respects to elders and communities past, present and emerging of the lands that the University of Sydney's campuses stand on. And for thousands of years, they've shared and exchanged knowledges across innumerable generations for the benefit of all. And we really have to thank them for why we're here in our progress. So I have a little welcome speech first before we kick off with our guest lecture, which I'm sure you're going to really enjoy. And I'm going to begin by just providing the background for this amazing event, the Coles Colloquium. And it was established in 2013. And it was really to recognise the contributions of the Coles family to Sydney Hospital and the SaveSite Institute of the University of Sydney. And this is because Sir Ken Coles had a number of roles. At the Sydney Eye Hospital, he was on the board for 22 years as a member. He was actually a chairman for 8 years. And in one of the publications, he was described as being sort of an elder statesman. He was thanked for his wisdom and his counsel. So he was a very pivotal person in establishing the SaveSite and in moving it forward. And he also served the university in many roles. He was a member of the University Senate, and this is a very privileged position. He was on the board, as I said, for SaveSite Institute, and he was the chairman of the advisory board. And he helped make SaveSite what it is today, and we were ranked 6th in the world recently. He was also not just helping here at the hospital and the SaveSite, but supported many charities. And he was a very generous man, generous with his time and also his support. Now, over the years, there's been a number of distinguished speakers at this event, and I've been to many of the Coles Colloquiums, and I've always learnt something new, and I've been inspired. I've been inspired to keep working towards finding better outcomes for patients with vision and sight disorders. So you can see from this list here in 2013, Professor Marie Bashir was the first speaker. We had Professor Bruce Robinson, Professor Paul Mitchell, Associate Professor Gordon Sanderson, Professor Mark Daniel, Professor Jane Latimer, Professor Frank Martin, who's here in the audience, Dr. James Mookie, Professor Robin Ward, Allegra Spender, who's MP, Professor Ian Constable, and then last year we were very honoured to have Her Excellency the Honourable Governor Margaret Beazley. Okay, so just a little bit of background about how we all ended up here. So in 1882, the Sydney Eye Hospital was started at Millers Point, and then it moved to Woolloomooloo in 1922. In 1964, before I was born, the Department of Clinical Ophthalmology and Eye Health was formed at the University of Sydney. And this was initially led by Professor Frank Claffey and then Dr. Eddie Donaldson. In 1977, Professor Frank Bilson, AO, was appointed as the Foundation Professor of Ophthalmology. And then in 1985, the SaveSite Institute was founded with seed fund from New South Wales ACT Lions Club Sight Foundation on the 6th of May. Moving forward in 1989, there was the Westmead campus established with Professor Paul Mitchell leading the Blue Mountains Eye Study. And then in 1997, we relocated to the south block of the Sydney Eye Hospital from Woolloomooloo, which is where we are today. And in 2009, Professor Peter McCluskey, who's here in the audience, became the second director of the SaveSite Institute and chair of ophthalmology. 2013, as I mentioned, was the inaugural Coles Colloquium. In 2020, we became one of the 6 flagship centres in the Faculty of Medicine and Health. Last year, the Snow Vision Accelerator was formed under the SaveSite. And then this year, I've become the 3rd director, and I feel very honoured and privileged to be here today. I'd like to thank Professor Peter McCluskey for his extraordinary contributions to the SaveSite Institute over the years. They're both in research, establishing the SafeSite Institute as a leading research centre, but also in education, with both national and international impacts. And some years there's over 100 students that take the postgraduate coursework of the University of Sydney. Clinical care, he set up a world-class uveitis service here at the Sydney Eye Hospital and has been very involved in community engagement, such as through the Community Day. He's not just been influential here in Sydney and nationally, but also internationally. His uveitis research has resulted in evidence-based treatment. He's won awards across countries, including in the Asia Pacific, the American Academy, and he's currently RANZCO president. So where are we in 2026? Well, it'd take another sort of full lecture to give a full debrief of all the activities at the SafeSite Institute. But just to sum up, we have professors, associate professors, senior lecturers, along with honorary and clinical affiliates who lead research here with global impact. And most of our researchers that are clinicians are appointed here at the Sydney Eye Hospital so we can translate those research findings into evidence-based care. Our Professor Claire Fraser from the SafeSite Institute has now been appointed the head of ophthalmology, taking over from me. And we have research such as the SafeSite registries led by Professor Mark Gillies, which is a unique global effort to collect real-world outcomes to inform practice. There's been leading corneal research. We got a Medicare item number for corneal cross-linking. Professor Gerard Sutton launched the spun-out Bianco from the SafeSite Institute, and we now have the Snow Vision Accelerator led by Professor Jonathan Crouston. Now, despite all this, saving sight remains a global challenge, and vision impairment is higher in Indigenous Australia, and it's predicted to double by 2020. Making saving sight a pressing issue. The costs to society are actually higher than stroke and mental health combined. And in the NHS now, 10% of all outpatient visits are ophthalmology, making it the busiest specialty. So we're privileged to be here, the SafeSite Institute at the Sydney Eye Hospital. And this is because we have aligned goals. Both of us want to improve the outcomes of patients with vision loss and eye disease. We have a collaborative leadership with Professor Andrew Chang, who's here in the audience, and Dr. Pauline Rumer, who couldn't be here tonight. Working together, we have complementary expertise in patient care, research excellence, clinical trials, and education, with many of our HDR students now registrars on the training program. So, and working together, we really can multiply and fast track our success. So just to end, this is sort of SafeSite Institute. We're launching into a new era. And as part of this, our vision will be continuing to transform lives really through saving sight. Our mission is really to enable breakthrough discoveries in ophthalmology through research that is multidisciplinary and collaborative. So that's working together. And the culture that I want to create is one of working together to drive innovation and connectivity, with values of excellence, collaboration, curiosity, and integrity. Now, we're very privileged tonight to have the Chancellor of the University of Sydney address the Coles Colloquium. And the bio of David Thodey is incredible. And I've just summed up some points here, but you'd be reading it again for a very long time because there's many achievements. But he has over 30 years of contributions to science, technology, business, and community. He's been involved in driving innovation, which is something we want to do here in startups, large companies, and public sector bodies. He's an advocate for the real world impact of research, which is something also that we're very interested in and moving forward here on this campus. He's passionate about environmental sustainability, being involved with the Great Barrier Reef, chair of Zero and Ramsay Healthcare, and an officer in the General Division of the Order of Australia for Ethical Business Leadership. I'd like to invite you up to the stage. Thank you.
Well, thank you, Stephanie. Well, thank you for that wonderful welcome. And it was just great to hear about Sydney Side Institute, I learned something. It was really great. Well, look, it's great to be here. And let me pay my respect to the Gadigal people, who are the traditional owners of the land at which we're meeting, and pay my respect to elders past and present. Well, being Chancellor of the University of Sydney, I think the most enjoyable thing I do as Chancellor is actually when I get a chance to come out to the faculties and the research institutes, because that's where so much happens, and it's always a real pleasure for me to come out and see what's going on. And it's just great to be here tonight with so many distinguished people, professors, doctors, business leaders, but people who make a difference in the world. And that's what it's all about, and I have a real passion for. And when I look at all the speakers that have been there, you realise, gee, what a wonderful group of people who have come here because they care, and that means an awful lot. And I, saw that Dame Marie Bashir was the first person to speak, and tragically we lost her this year, but she was such a big contributor to Sydney University as well over many years. So it's sad that she's gone. And Rowena, it's just thank you for you and your wonderful husband. As you went through what Ken had contributed here, I think I met him at the Warren Centre, because I've sort of had an engineering and technology background, and he was a director there. But what a wonderful contribution he made to public life, and he did it in such a wonderful way. And then, the contribution here has just been incredible. So wonderful to see you as well. So, what do I want to talk to tonight? Well, it's something that's sort of pretty deep in my psyche and what I've been involved in for 30 or 40 years, and it's a challenge we have in Australia, which is fundamentally, how do we take all this wonderful research and actually make an impact, which we do here at the Sydney Institute. And of course, eye health is such a critical example of what we have done. When you get great focus, you can really make a difference. So what I want to talk to you about tonight is talk a little bit about our health system, because I see it through the eyes of Ramsay. It is private hospitals, but they're okay. And I want to talk a little bit about some of the changes that I think we're all seeing in healthcare, and it is happening at a really rapid pace. And then try to address a little bit of this challenge about, why are we so good at discovery in Australia and yet not great at translation? And what is it that's really driving that? And is there anything with it we can really do about it? I will sort of pose a way forward as we go through, but I think I've got 20 minutes. Is that right, Stephanie? And we'll see how we go. Well, let me start with the good news, because we don't say it enough. I think we have built one of the finest health systems in the world, and we don't actually recognize it enough. It is actually a very, very good health system. We live longer than most people in the developed world. We're now, our average lifespan's up around 83 years. I think the average is about 81. We have some of the best survival rates. So if you have a heart attack or a stroke, and we don't celebrate that enough. And interestingly, because I have to look at it, we're actually pretty efficient at delivering healthcare. When you look at the United States about how much they spend on healthcare and healthcare per head of population, they are way over what we do here in Australia. So we're pretty efficient, and we deliver a great public health network, which is just tremendous. And as I really alluded to, our medical professionals are considered, I think more than considered, they are some of the best in the world. And that's from our researchers, clinicians, surgeons, GPs, researchers, nurses, and the administration, because you need good administration in healthcare. But, and this will run through everything I say tonight, that success is not free, and it's not guaranteed at all. The very fact that now we have this great survival rate from cancer, stroke, and heart, and we live longer, is placing pressures on the system. In fact, our success, in a sense, is becoming an issue for us, because as we have an aging population, it's putting more pressure on the system. And I think it is absolutely critical that we maintain these high standards of health care as we try to keep our populations healthy and contributing. But I do think it is going to depend on how well we invest in research and how we fund universal health care in Australia. And we must keep investing in research, translation, acute care, aged care, and also post-operative and preventative health care, because these are the things that will create a strong nation. And as I said in my introduction, medicine, I think, is going through an enormous set of changes that you will see more than I will. But there are two key drivers that I think are quite unique in terms of how we're delivering healthcare. And the first is, as you know, we are becoming more and more digital. And that's actually how I ended up in being involved in healthcare, because I've been a technologist really all my life, worked at IBM, started in engineering, and then have seen the impact of technology. And so much of healthcare is now driven by technology, and now AI is sort of touching nearly every part of the health system, from diagnostics, the Da Vinci robots that we use in our theatres, administration, drugs, touching nearly every part of medicine. The one thing I will say, I'm very glad that AI is not touching, is judgment and empathy, and that is something that we need to make sure is always the case as we go forward. And of course, data is guiding all our decisions, and big data is giving us the ability to analyze and make better diagnosis and actually deliver better healthcare than ever before. And of course, the other big trend that you would see is that we being able to deliver is now really personalized medicine. Now, I want to be very clear, this is just not personalized drugs. This is what we're talking about, a new generation of really tailored care to the individual, real time and on, in the moment, and, from screening, treatment, monitoring. And it is to the individual, that down to their genetic level and also into their specific requirement at that moment. This is an enormous change, and you are delivering it here at the Institute. And that's what we need to see more of, because ophthalmology is one of the most data-rich, most technology forward-looking fields of medicine that I have come across. And considering how fast it's moving, we've seen macular degeneration was, just untreatable a few years ago. Incredible. And this, it has now been an approved treatment. Inherited blindness is being met with gene therapies delivered directly into the eye. And artificial intelligence is reading retinal scans, and you're doing it here, which is just incredible. And now the wonderful smartphone is able to do, eye examinations just on the moment. Now, who would have believed it? But you can do your blood pressure as well if you need to. But all of this is underneath, this is sitting data. And that's why I'm so excited to see what the Save the Site registers are actually doing, which are being built right here, because I think they are one of the largest in the world, if I have heard correctly. And it's about outcome data sets, which is really very, very exciting. So this is sort of really pervasive in healthcare, and I'm seeing it at Ramsay Healthcare now. I mean, we are a private hospital group, and so we do a little bit of research. We're caught with the Paul Ramsay Foundation as well. But I'm seeing personalised mRNA cancer treatment today, and we're doing it in Brisbane, and we've got a ward up there. Also, we're doing immune cell treatment as well across in Perth. So these are things that are happening very, very quickly and going from research right into the core of acute healthcare delivery. I find it amazing. I mean, I feel inspired by what I see. I feel inspired by the people I meet working in this area. However, one of the big challenges is that personalized medicine is not cheap. And the big challenge is, how do we get it to more people? And who is going to pay for it? Because this is the challenge of delivering healthcare to everybody. Now, I am an optimist, I think. For those who have been in the technology area, there's a thing called Moore's Law about the advancement of technology and the cost decline, which we're sort of seeing if you follow technology in NVIDIA and this wonderful, AI chips. I think the price of technology will come down, but that doesn't mean that it ends up being accessible to the wide community. And I think that's something that we really need to work on as we move forward. Now, as I said, I think as a nation, we are great at discovering, and our medical research is generally world class. I think we're now rated 7th or 8th in terms of health sciences. SSI sits right up there at the top. I think you're, sitting alongside the University College London, Harvard, Johns Hopkins, etc. So you're doing well. But hold that thought, because it really sets up the hardest question that I struggle with. If we're so good at discovery, why do we struggle to get the benefits from it? What is it in our system that is preventing us being at the high end, being one of the world leaders in that translation? We have this, great research and center. It's not just in medical healthcare. I could go to engineering, I could go to robotics, I could go to any, biomedical science area, and the story is the same. It's an interesting one, because I'm not just talking about commercialization. I'm talking about translation to value. And when you look at the scores of how we're rated as a country, we just don't get there. We just don't get there. So let me take you through it. And I should, before I do that, just I think it was Tim Ayres, Senator Tim Ayres, said the other day, when he was talking about this translation capability, he said, the sad truth, and giving the government's words, is that we are virtually giving away our world-leading research to others to commercialize, which I find absolutely staggering. So the numbers are sobering. So our total investment in research over the last 2 decades has declined enormously. We are now at 1.68% of GDP. OECD average is about 2.4% of GDP. It's shocking, absolutely shocking. Even worse than that is that when you look at government expenditure compared to business expenditure has declined more by something like 35% over the last decade. So the 1.686% of GDP, about 1.1% is government expenditure and the rest is business, and that's declined enormously. And when you look at what we talk about in terms of economy, because R&D relates to economy and economic complexity, we rate at 105th out of 300 in the world. And guess what? The country that beats us is Botswana, of all countries. And when you look at the data, it's an interesting one, because you start to see things like, we're not seen as collaborators. And in the OECD, the one score that really stands out is the collaboration between business and university. We're at the bottom of the top 33, business and university or academia. And it's strange to think, why is that? Why is it that there is not a sort of a true engagement? Maybe we need more business people speaking here. Maybe that'll do. Also, I think we've got a cultural problem. Australia, which I don't think is risk averse, but we are seen as being very risk averse. And I think we confuse risk management with risk avoidance. And we are not willing to step up and give things a go. And yet what is in our culture as Australians? It's a give it a go sort of culture. And yet in this area, our tolerance is just way too low. And so this low trust between different sectors is not good. And I would also put in there, it's government to academia to industry where we really have a problem. And, I see it in so many areas. I mean, you look at the area of clinical trials, where I think we're now We do roughly 4% of the world's clinical trials, and we should be, with the amount of capability we have here, we should be up in the 6-7% range. And if we were to do that, we would start to build a whole new set of capabilities in this country. And we haven't got a funding model that's kept pace. I mean, I've seen, robotic surgery, and Da Vinci robots in operating theatres, but they, just has not been a consistent investment in the use of that wonderful technology over the last decade. And you can apply that to genomic and cell-based therapies. So we need to find a new funding model. And one thing I have learned in healthcare, you need to follow the money. And because without money, we can't get things done. So let me be very clear about one thing as I talk about this. None of this is a failure of talent. In fact, I look around the room and I see wonderful people. It is a failure of our system. And that's where we need more thinking and more engagement. And you need it consistently over a long period of time, not just sort of a shot in the arm. So let me just sort of deviate for a moment, because I think the answers are pretty simple, actually. For those who may know, there was a report done by Robin Denham, who you're aware of, who was asked to look at the whole research and innovation ecosystem in Australia, and it was called Ambitious Australia. By the way, we've had a few of these over the years, and I've been involved in a few of them myself. But Dr. Fiona Wood, who many of you would know, was involved in that, as was Professor Ian Chubb, and they issued their results in March. And I actually think they did a pretty good job of just articulating how you change a nation, how you build capability that can bring people together. So let me just quickly share with you the 5 sort of principles that they identified. And I actually saw on your charts many of those, Stephanie. So first of all, and this sounds simple, but it's actually very hard, you need a shared long-term national vision. You actually need to say, this is important to this country. And it needs to bring all the elements of Australian R&D together. Now, anyone who's been to Canberra knows that it's a very disparate, uncoordinated group of people. R&D funding flows all over the place, and half the time you have no idea where that funding is going. So they've recommended there should be a National Innovation Council to try to bring together all the different sectors to have one sort of view about what we're doing. Again, in the Westminster system of government in Australia, that is actually very hard to do. Secondly, you need to fund the foundations, and the foundations are great academic standards, great research, and we need to do that. And yet you look at our funding of R&D, it's going the wrong way. And you think, what? Why is this happening? So we need to invest in academic excellence, because without great knowledge, there is no innovation. Thirdly, as I talked about before, we need collaboration. They talk about a term called intentional collaboration. That's going out of your way to collaborate, going out of your way to make, a connection with somebody else who may not be in your particular domain. I've looked over the years at, the Swiss model, the Swedish model, the Scandinavian countries, very good, the so-called triple helix model of bringing government, academia, and industry together. Very purposeful in driving real collaboration. And if you look over the years, that they've been very, very successful in doing that. And the Denham report does pick up on that. Fourthly, they talk about a relentless focus on translation, and the words of the report, it says turning knowledge into goods and services that transform the lives of people. I mean, it sounds simple, doesn't it? I remember when I was chair of CSIRO, one of the great things that CSIRO did, which many of you will remember, is that they had the patent for Wi-Fi. Remember the story? Actually, it was part of Wi-Fi. It wasn't the whole of Wi-Fi. Anyway, we'll take the credit for it. Anyway, we through that patent, we were able to generate $300 million of recognition, which was great. The great thing that the government allowed us to do was to reinvest that money into a group called Main Sequence Ventures. Have you heard of Main Sequence Ventures? Main Sequence Ventures is a venture capital arm that was specifically focused on getting research from the lab into beta tests, nothing more. So they invested in that company Q Control, which was quantum computing, which was about those little qubits, managing the qubits using software, and have been incredibly successful. But it was a very focused bit of effort. And that's the sort of thing we need to do more of, because if we can get that money into those sort of core areas, then we can really make a difference. And I could go on and talk about clinical trials. I'm not convinced that R&D tax incentives make all the difference. I think it's more about attitude. And what I worry about is that we don't have a culture where we celebrate innovation. I think we can get a culture right when we can talk about our best researchers, and it comes to mind, so a primary school teacher or a student can remember a great researcher as well as they remember a great sports person in Australia, maybe we would have arrived. So we need to get a culture where we celebrate and recognize and put people on a pedestal, well, maybe not a pedestal, but just give them praise and adoration far more than we do today. Because I think this is what's really important. And I think that the Denham report is very good. However, again, it goes into Canberra and unfortunately it may be put in the bottom drawer, and we need to keep bringing it to the fore. So, just so you know, there were 6 industry areas that they talked about in that report. And one of the most important one was health and medical, and requiring big research. And of course, we do have the reconstruction fund that is there, and we have the medical research future fund as well. They are starting to release more funds from. So I think there is an opportunity here, and I really hope that we continue to step into it, because the future of medicine is personal and it is digitized. And I think that, as you've seen, we do need this vision as we go forward. Another report was done by a guy called Bill Ferris. Does anyone know Bill Ferris, who used to be at Innovation Science Australia? And he did a report when Malcolm Turnbull was Prime Minister. And I really like where he got to, because it was a wonderful report. It sort of talked about all the things we should be doing. But his vision was to make Australia the healthiest nation in the world. Now, just think about that. It wasn't, yes, because we love the outdoors, we love sport, but it was to make this the healthiest nation in the world. And that would drive, great research. It would actually put real focus on where we're really good. Anyway, it sort of got a little bit of momentum, but didn't quite get there. But if we can do that, I think we will improve our innovation. We will improve the lives of Australians. And I think it comes down to leadership. And that's why I stand up and I talk about these things, because I think they're important, because it has a big impact. So let me finish with this, because I think we may not be able to change Canberra, but what we can do what we can make a difference at. And that's here in Sydney. And the University of Sydney does have a big vision for medical research. We have a big vision for both the university, but for the city of Sydney. And we want to make a difference. We want to be an engine room of discovery focused on improving people's lives. And that involves a university being outward looking, being willing to step out and be more inclusive, being able to work with industry, be able to work with government more than maybe we have done before. I mean, as you know, the university already has extensive research centres right across the university. I mean, we have the big ones, Charles Perkins Centre, we've got Susan Wakel Centre, you've also got the Brain and Mind Centre, got this institute, but there's 60 of these spread around the university doing incredible work, and we need to get more of them as we go forward. Because if we are going to have an ambition to be a global, a world-class medical research centre, we need to build strong partnerships across all universities, across all research institutions. That includes the private ones, Garvin, Kingsmill, Victor Chang as well, and with industry and with government. And of course, the Sydney Biomedical, etc. Has anyone been out to the university? It was a big hole, and now it is starting to rise. We were out there just the other day. I think they're up to about the 5th floor now, and it's starting to take shape. But what a wonderful, wonderful opportunity we've got there, which is that it is a physical building, but it's connected into RPA, it's connected back into the university, and it's going to be a place where we can bring people together, who are doing our researchers and clinicians working across industry. So we've got people who will be working in, oncology, cardiology, neurodegenerative diseases, in a core site. But it's a group of engineers and researchers all coming together, multidisciplinary research that we think can really make a difference. Now, buildings don't change the world, people change the world. But so we need great leadership in terms of bringing them together, finding, problems to solve. And I'm very encouraged with some of the people now who are coming in there, because it's this type of collaboration I think can really make a difference. But, we need each other along the way. And therein lies the challenge, because we need to be in the partnership business. But, I remain incredibly excited about the future opportunities, about what we can do. And I just want to say the Sydney Site Institute is such a great example of what is possible. And I think if we can do more of this, we can achieve, outstanding things. And I remain very committed to that objective. And just maybe, we can be the healthiest country in the world. So thank you very much, and look forward to seeing you. Thank you. Oh, I'm very
Thank you very much. There's a lot of food for thought, and I think it's really along the path of where the Safe Site Institute wants to head, with the Sydney Eye Hospital working together, working on innovation, building partnerships. So thank you again. And okay, so I'm going to, yep. Thank you very much. I'd now like to invite Professor John Grigg, who's a professor here at the Safe Site Institute. He's a leader of the Eye Genetics and Inherited Retinal Diseases group, who also consults here at the hospital and at Westmead. Subspecialties include glaucoma, cataract, pediatric and genetic ophthalmology, and clinical electrophysiology, which I think we're about to hear about. And he undertook fellowships in Australia and the UK, in Manchester, where I did as well. And his research interests are genetic eye disease, glaucoma management, and electrophysiology of the visual system. So thank you, John.
Thanks very much, Stephanie. Look, I also acknowledge the Gadigal people in the lands which we meet today, but I'd also like to acknowledge to start my great appreciation to both Ken Coles and Romina Danziger, who've been great mentors and provided wise counsel over the last quarter of a century. So part of my presentation will overlap some of the things that they've contributed in this area. So I wanted to talk tonight about visual electrophysiology, which is one of my interests, and how it relates to solving problems and mysteries in vision. So really just to start with, what is visual electrophysiology? And it's really a series of standardized tests to assess visual function. But who does it serve? And really there's these groups of people where it helps provide an objective measure in children where children can't tell you what they can't see, or we've got people where there's sights failing, but all our standard assessments in the ophthalmology rooms can't work out why that's happening, or where the diagnosis needs to be confirmed so we can start or monitor or change therapies, particularly immunosuppression. We have groups where there's autoimmune retinal conditions, where these are insidious conditions which present slowly, and often there's no other signs, and it's only electrophysiology that can find the answer there. Then there's a subset of that where there's cancer-associated retinopathy, where we can have an occult malignancy and their first presentation may be vision impairment. Then there's the large group of genetic eye disease where we need to get accurate phenotypes so we can help our genetic colleagues to work out what the genes are that causing the particular disease. I'd like to sort of step back and sort of say, well, how does electrophysiology relate to vision? And there's a question that was posed to me a little while earlier this year was, well, what is vision? As ophthalmologists, we all have an idea of what vision is, but how would you define vision? And really, on one level, we can talk about vision as foundational to what it means to be human, to learning, connection, independence, and identity. But on the other hand, it's how we try and then measure that. And vision is not just one measurement. And I'd like to use the analogy here of the Indian parable of the 6 blind people who describe an elephant. So some of you may remember that, where the elephant was assessed by these 6 people and somebody grasped a tail and thought it was a rope, or the trunk and it was a snake. But all of those together gave you little bits and pieces of the picture. And in the parable, it's really looking at more overall in life and so forth. But we can apply it to vision, where we have these measures, which then sort of give us only a part of the insight into what vision is. And yet the elephant is really what the vision is. And so when we come to sort of looking at applying vision here, we can actually conveniently break down vision to sort of 6 main components that make up the vision that we can then measure. And so this could be the spatial arrangement. So that's sort of what we use with visual acuity. The contrast, how do we detect between different shapes and shadows? Or the extent of the vision, the visual field, so perimetry. Adaptation, meaning, how we can see in the dim light to the bright sunny day at the beach. Or we can see colours with the different spectral areas. Or temporal kinetics, so dealing with motion, how those change. And so it was useful sort of when we were hunting and gathering, spotting targets, or now sort of watching movies, all those things. But those 6 different domains go to make up what we see, yet there's no one spot in our brain that we have our final image. It's drawn from multiple areas. And so I wanted to sort of then take that knowledge we've just talked about and look at how we might assess vision. And I've put this together as sort of 2 different domains, sort of a y-axis, which looks, can you fix on the target? And so then we can use tests that look at that way, or you can't. And then looking at how we assess things across different light levels, some from the photopic or very bright sunny days, mesopic, which is a room lighting like here, or in a dark, starless night, or when there's no light at all. And onto that coordinate map, we can sort of think about how our tests that we use commonly in the clinic fit together. And from that, we can then see where visual electrophysiology might fit in. And so we have where we need fixation, we have sort of the standard visual acuity, the chart that you've all used to reading about. We can do that in dimmer light or putting filters in front, or we can use color vision, contrast sensitivity, all those fit in that higher needs fixation. Then in the middle sort of area, we've got measuring the extent of the visual field. And one of those areas is the visual evoked potential, measuring the pathway. That's part of the electrophysiology. And then one that's been developed here, the multifocal visual evoked potential, I'll talk about in a moment. And then you can see, so there's an area of electrophysiology there. Then we've got where you can't fix, you can use electrophysiology as well, sort of measuring the retinal function. Or if you've got really almost no vision, then we've got this full field stimulus test in the other corner here. So you can start to see where our tests come together and where electrophysiology fits in. This is sort of an overview of what we can do this is a structure of the retina, and we have our inputs in the top left-hand corner there by applying different stimulus, which may be patterned or flashes of light. They're then captured by the light receptors, and using different parameters, or the strategies we talked about earlier, we can then isolate the individual photoreceptor cells or the inner retinal cells, or look at the whole pathway, and we get the patterns that we see there on that. So that sort of gives us an overview. What I'd like to take next is look at the relationship that visual electrophysiology at the Saison Institute and Sydney University has had with one of our large basic science groups at Sydney University. So that's the visual physiology and neuroscience. And I've put together some timelines here. On the left is sort of the role of visual physiology, which Sydney has been extraordinary track record on this. So Peter Bishop, who started this in the 50s, many of us learned in our basic science exams, he really described how the mechanism of binocular vision works, so stereopsis. Then followed by Jack Pettigrew and Sefton, And then coming through to Bogdan Dreyer, who looked at different parts of how the pathways sends information in parallel pathways. Then to Paul Martin and Uli Grunert, who are part of SafeSite today, who really identified how the blue pathway works. Then Dario Perotti looking at inner retinal cells. And so that's sort of the basic science group there. And then on the right-hand side, when Frank Bilson came, he then recruited Vagan from the UK, and we established the first electrophysiology service in Australia here. And then we've had some great leaders who've gone on to better things. Brett Jeffrey in the early 90s now heads the service at the National Institute in Washington. Then in the mid to late early 1990s, we recruited Sasha Clistorna and Stuart Graham, who developed the first multifocal visual evoked potential, which is a way of objectively measuring the visual field. So without having to press the button, you can record this in the brain. Again, that evolved from going from a glaucoma approach with Claire Fraser starting to go into optic neuritis. And then we had this area, I just throw this in the middle there, of the Coles-Peterson-Clistorna group, because the original multifocal VP was commercialized and went through FDA approval, but then it faltered. And then through Ken Coles and Chris Peterson, with Sasha Clistorna, they resurrected into vision science. And I'll talk about that a little bit more. We then had the experience of having Graham Holder, who headed the unit in London, came and became an honorary professor here and guided us. And just this month, we've hosted the 63rd International Electrophysiology meeting here in Sydney, which had a record attendance for the course and the program. And the team here have been involved in developing new standards for electrophysiology to help better provide that sort of precise vision or precise assessment of vision, and that can be translated to multiple different groups. The last slide, which is sort of quite diagrammatic, but really I wanted to add governance to this, and that's particularly for this sort of audience, bringing in the SafeSite governance program. And we can see that it was founded in 1985 with Frank Bilson, the first director, then Peter McCluskey, then now Stephanie's taking over this last week. But the board presidents, and I wanted to acknowledge here Ken Coles, who had been on the board to help sort out the finances before he became president of the board. And that 2001 and 2012, before the SafeSite Institute changed from being an independent board reporting to the Senate, then became a complying center of the university. But Ken, through his leadership, identified that there was an opportunity when the previous commercialization of the multifocal VP was faltering, that organised for the university to take back the patents. And then he did this through a board subcommittee, which I was fortunate to be on with Peter McCluskey and Sasha Clistorna. And that then led to Ken and Chris forming this company called Vision Search, which then was able to rewrite the software, take the patents, and then develop this system, which has then become the standard for clinical trials in assessing demyelinating disease with multiple sclerosis and optic neuritis. And that's been a huge success story that's allowed new therapies to develop for multiple sclerosis, and that's been used internationally around the world in those outcome measures. We now have the electrophysiology service here sort of supplying all the different units in Sydney Eye Hospitals, because there's questions that every part of the subspecialties of ophthalmology has, so neuro-ophthalmology for ocular plastics with thyroid eye disease, where the optic nerve is affected, or the corneal unit looking before they do their corneal grafts, which Stephanie's involved with. You need to see that the retina is still working, or looking at glaucoma. We've worked with the genetic eye unit led by Robin Jamison at CMRI to really help provide the phenotypic background for their genetic work. The inflammatory eye disease led by Peter McCluskey and the team there. It's really important to try and help monitor therapies and also to try and differentiate those autoimmune disorders. And then providing some of Mark Gillies' work with Macular Research, particularly has been involved in the MacTel, which is another success with the Lowy Foundation's funded here. And more recently, what Matt Simunovic, who we'll hear from in a moment, and Dario Perotti are looking at optogenetics, and then just in general pediatric trials. So this last one shows to bring together how the electrophysiology has cross-fertilized across the basic science of physiology into understanding more about objectively assessing the visual system and then how it's used in the clinical service. So it's been a great privilege to be part of that journey and to present here tonight. So thank you.
Thank you very much. And so I now have the privilege of introducing Professor Matthew Simunovic, who's a professor of ophthalmology here at the SafeSight Institute, and he leads the Retinal Disease and Rescue Group. He's a consultant surgeon here at the Sydney Eye Hospital and Sydney Children's Hospital Network, where he specialises in surgery and diseases of the retina. His research interests include emerging treatments for retinal diseases, experimental vitreoretinal surgery, and visual psychophysics, which sounds all very exciting. He was the first Australian ophthalmologist to be awarded a Career Development Award from the Foundation Fighting Blindness of the USA. So that's that's a real privilege. And it's supported by major grant funding from the Macular Diseases Foundation and the NHMRC. Thank you, Matthew.
Thank you very much, Stephanie, for the kind introduction. Generally speaking, psychophysics puts people to sleep. I'll try not to keep you too long from your canapes. So this is a bit of a whistle stop through vision restoration in retinal degeneration. Why is it important? We know that inherited retinal degeneration is the leading cause of blindness in kids and adults of working age. Age-related macular degeneration is the leading cause of blindness in people of retirement age. We know a lot about these conditions, and our knowledge of these conditions indeed has improved over the last couple of decades. We recognise that no single approach is going to be suitable for restoring vision in all of these patients. I'm going to be talking about 4 key pillars in developing new treatments for retinal degeneration. They are the assessment of function, which has been covered well by John already, so I won't labour that point. I'm talking about rescuing visual function in cases where there are sufficient viable retinal cells. Where this is not possible, I'm talking about repurposing retinal cells through optogenetics. And finally, I'll finish up with replacing retinal cells via either cell therapy or electronic retinal implants. So the main business of vision arguably is sort of spatial contrast detection. So under many light levels, the visual system is designed to extract contrast from the visual scene, as it were. And it's for this reason that, by way of example, if you read a letter chart through a pair of sunglasses, you'll do just as well as if you're not wearing the sunglasses. And equally, if you do a visual field test, you'll do just as well through a pair of sunglasses. And why is this important? We've been able to demonstrate in Sydney that the early stages of retinal degeneration produce pretty much the same effect on your visual function. So although your vision may appear to dim, it's not picked up with conventional clinical tests. And for this reason, we need specialised tests. And how do we get around this? As John has alluded to already, we need specific paradigms, and we need to test under different light levels. And this is really underscored by a test which I've got on the top right, which is called a multiple luminance mobility test, which was developed for assessing the only currently available retinal gene therapy, Luxturna, which I'll talk about very briefly. So essentially, it's like a little obstacle course that you run through under different light levels. And in the phase 3 trials, they were able to demonstrate that patients were able to navigate this little obstacle course under far lower light levels. So it's really important that we have adequate measures of visual function over and beyond our standard clinical tests, as John has already alluded to, in assessing these new treatments as they become available. So when there are sufficient viable retinal cells, it may be possible to rescue vision in some instances, and I'm going to just limit this to talking about Luxturna gene therapy, which is the only available retinal gene therapy. It's delivered via a subretinal injection. I've got a looping video of a subretinal injection of Luxturna. So it's 0.3 of a mil of a solution that costs $500,000 per eye. It contains a viral vector, so a little virus that has a genetic code for the correct copy of a gene called RPE65. That gene codes a protein of the same name. So that protein is expressed in the supporting layer of the retina, the retinal pigment epithelium, and it's responsible for recycling the version of vitamin A we need to detect light in the retina. So in patients who won't produce this protein properly, they have greatly impaired light sensitivity. If they then undergo Luxturna gene therapy, this light sensitivity greatly improves by just over 100-fold. It was demonstrated in the phase 3 trials, and this is borne through to real-world studies, including those in Australia. As yet, no other retinal gene therapy has achieved regulatory approval. So it's becoming increasingly recognised that surgical delivery is important in delivering these expensive treatments. Initially, it was assumed that if you standardise the steps of surgery, most of the solution would end up in the subretinal space. But data from several years ago in animal models suggested that reflux of these therapeutics can be significant, up to 96%. So if you're going to be injecting a drug that costs $500,000, you want most of it to get there, not just 4%. We've developed a technique in Sydney to assess how much therapeutic solution ends up in the subretinal space. It's shown at the top right. I'm performing a subretinal injection of a clot-busting medication called TPA. It's been mixed with a fluorescent salt, which we use in the clinic all of the time, called sodium fluorescein. And you can see qualitatively what happens when you perform this injection in a patient who has a submacular hemorrhage from age-related macular degeneration. So it spreads throughout the subretinal space, but also some of the drug refluxes. And so at the end of the surgical cases, we've been able to take all of the fluid from the eye, and we can measure the volume of this fluid, but we can also determine the concentration of fluorescein using fluorophotometry. And this tells us how much drug has been refluxed. And we've been able to demonstrate that in humans in vivo, it's around 4% on average. And we've also performed a randomized trial to prove the preferred way of delivering such therapies, which we published recently in American Journal of Ophthalmology. So there are limits to rescue. There are more than 300 genes or loci that have been implicated in the pathogenesis of inherited retinal degeneration. Of course, age-related macular degeneration is multifactorial. So if we don't have a specific treatment such as Luxturna for an inherited retinal degeneration, it may not be possible to rescue vision, at least by that approach. Also, it has a use-by date, so if there are no sufficiently viable retinal cells to rescue, there's no point in trying this approach. We need to turn to other approaches, which I'll be talking about now. Optogenetics to repurpose retinal cells, and other approaches to replace retinal cells. That's cell therapy as well as electronic retinal implants. So what is optogenetics? John presented a diagram of the retina. These are the photoreceptors, the light detecting cells of the retina. So the final common pathway to losing sight in retinal degeneration is loss of those cells. But the relay cells that connect the light detectors to the parts of the brain involved in vision, the bipolar cells and the retinal ganglion cells, they persist, but they're not normally light sensitive. So optogenetics is a form of gene therapy in which you express light sensitive proteins in these relay cells, and they therefore become new or novel photoreceptors. And it's been proven to restore vision both in preclinical as well as translational models of treatment for inherited retinal degeneration. So inherently, people thought in the early days of optogenetic approaches that the more protein or light-centered protein you produced in the retina, the better the vision. But we've been able to demonstrate that if you sort of express a lot of protein indiscriminately throughout the retina, it actually can create a lot of noise. And therefore, this is not the preferred approach. You need a targeted approach to specific retinal cells. This is compounded by the fact that the degenerating retina produces a lot of inherent noise by itself. And we've been able to also show in Sydney that if you repurpose commonly available anti-inflammatory medications, you can suppress this inherent noise and potentially improve vision when you deliver optogenetic gene therapy. So cell therapy is on the horizon. There are a number of reasons why it may work, one of which is replacing lost cellular populations. One of the difficulties over the years in cell therapy has been identifying a plentiful source of cells for therapy. In Sydney, we've been able to demonstrate that the adult human retina actually contains a niche of progenitor cells, cells that can turn into other cells within the nerve layer of the retina, and we can get these when we do retinal detachment repair. I've got a video at the top right of me trimming a flap tear during retinal detachment surgery. If we then store these little explants, these little fragments of tissue, then progenitor cells will emerge from them, and those can restore sight in preclinical models of retinal degeneration, and that's something that we're exploring here with colleagues. Finally, electronic retinal implants. So the proof of concept of these came from the 1960s from work that was performed in Cambridge by Brindley and colleagues. And 2 different electronic retinal implants have previously been produced, which were approved internationally. They're no longer produced. They were large, they were cumbersome, they were connected to external battery sources. But there's now available a sort of next generation implant, if you will, which was approved last week in Europe for treating patients with advanced age-related macular degeneration. I've got a surgical video of me implanting one of these in Sydney in a patient with inherited retinal degeneration in a clinical trial called Primalia, which we're conducting at present. This is a 2mm chip, and it's different to preceding implants insofar that it acts more or less like a solar cell, so it doesn't need an external battery source. It's charged by laser light, which is projected onto it. It's a 2x2mm array. It's got just under 400 electrodes. And so the laser light also delivers the image, and that's all delivered via a special pair of glasses. They're rather cumbersome at the moment, but the company has plans to convert it into a pair of glasses that look similar to smart glasses. And so it passed through pivotal trials last year, and the data were published in the New England Journal of Medicine earlier on this year. As I said, it's been approved for therapy in Europe, and it works by stimulating, as it were, residual nerve cells in the retina. So we've seen that no single approach will be suitable for all of our patients with retinal degeneration. We need to have targeted approaches. We need specialised tests of visual function. If there are sufficient viable retinal cells, it may be possible to rescue them. If this isn't possible, then other approaches may be appropriate, including repurposing through optogenetics or replacement through cell therapy or electronic retinal prostheses. Thank you very much.
Thank you very much. That was some really cutting edge surgery there, and I'm just glad all the corneas were clear enough for Matthew to be able to see through to do that fantastic surgery, which is what we do here as well, keep those corneas clear. So I'd like to now invite you to refreshments and networking. The balcony is closed and strictly off limits, so please don't go onto it. It may fall down, I'm not sure. The bathrooms are located to the left of the stage, and the lift is located past the foyer on the right-hand side. And there's some fire exits around that you can head to in the event of a fire, hopefully not. And the event tonight will end at 8 p.m. sharp. But I'd just like to thank you all for coming. It's great to see so much enthusiasm in winter on a Wednesday night to come and hear about these exciting, the exciting future we have ahead, really, in the SafeSight Institute at the University of Sydney, being located here at the Sydney Eye Hospital and working together. And also really like to thank the Coles family. And Rowena, thank you again for coming. I've always seen you here every year at this event, and it's a great privilege to catch up and also for us all to be reminded of the role Ken had in really bringing the SafeSight up to the next level. So thank you, everybody. And thank you. I should also thank, last but not least, my team, Susan and Cami, that have been working very tirelessly on pulling all this event together. Thank you.